Okay, so the next part of transporting animals is 9.2, the heart, and this is part 1 of the video. The first objective we're going to look at is to name and identify heart structures, such as these. The first one is the walls, and they're made of cardiac muscle. Now, cardiac muscle never gets tired, always contracting, you don't really control it. There's chambers, such as the atria and the ventricles, and they have different thickness of walls because some have to pump blood further. The septum divides the left and right side of the heart. It runs down the middle, and it's muscular, and it separates oxygenated and deoxygenated blood. So fetus hearts have a foramen ovale, which is a hole between the right and left atrium. This is because blood needs to bypass the lungs, because there's no need to get oxygen from air, because there is no air in the womb. The infant, the fetus would get oxygen from the mother, so it doesn't need that pulmonary circuit, and so blood just travels around the systemic circuit to the body. At birth, this hole closes, and blood then travels to the lungs. A hole in the heart is when that hole doesn't close, so they look slightly blue when they're born. Structure now. The chambers of the heart, you've got the atria at the top and the ventricles at the bottom, and then you've just got to remember that right is left and left is right. The valves we've discussed already, the 4 valves: right AV valve, left AV valve, and the semilunar valves. The coronary arteries are arteries on the outer surface of the heart, just here. These are the coronary arteries, and they supply the blood, the heart, with blood, and as such oxygen and glucose for respiration. This is because the heart needs to contract and respire. Tendons, these hold AV valves in place. When the heart contracts, tendons are pulled, which pulls the valves open and closed. They're not on the SL valves. These use flaps. These use tendons to help them close. Again, a picture of the heart. You've got the oxygenated side and the deoxygenated side. We've discussed the flow of the heart already, so I'm not going to go through that in too much detail. But here are the tendons here holding the AV valves in place, and when this contracts, this muscle here, this will pull these tight and closed. So then the blood must go through these flaps here, which are the SL valves. So this part is about the heart dissection, and it's all about identifying structures as you dissect a heart. So the first thing you do is wash the heart, and then you observe the surface, and you're looking for those coronary arteries, which are just here. And the left, the ventricle is thicker, so you would look for this thicker muscle, and you can do that by feeling it or just observing it. Then you make an incision from the base of the left ventricle to the left atrium. So that would be here, from the base of the left ventricle up to the left atrium. Remove any blood clots. You'd want to identify tendons. You'd see these as little white strings, just here, little white pieces of strings, and just here again as well. And then the chambers, which are here. You've got the left atrium here and the ventricle here. And the ventricle is much larger area and has a lot more thick muscle, whereas the atrium muscle is very thin. You've got to find the AV valves, the tricuspid and bicuspid, and you've got to be able to say that they have tendons. So here's the valves here, the AV valve between them two, and the tendons are just coming off those just here, and they attach to the wall of the ventricle. Okay, so the last thing you want to do is find that the aorta is thicker than the pulmonary artery, and this is the aorta here, and you can see that it is a lot thicker than the pulmonary artery here. Okay. The next objective, to state the direction of the blood in arteries and veins, we've already covered, so this is where you'll find that information, and I'll leave that there. We've also identified the valves in the heart, so I'll leave that there. The next thing we need to do is explain the difference in thickness between the left and right ventricle walls. Atria contract first, and then the ventricles. Atria, or atrium for singular, have thin walls, and they receive blood from veins with low pressure. This blood travels through relaxed atria and AV valve straight into the ventricles, but when the atria contracts, the final bit of blood flows down to the ventricles and it's a short distance, so there's no need for these thick walls. So the blood, as it comes through the vein, pulls in here, starts to trickle through to the ventricle. As this contracts lightly because of the thin walls, it pushes the remaining blood down to the ventricles. The ventricles have thick walls and they receive the blood from the veins initially, but then from relaxed atria as they contract. They pump the blood to the lungs or the body through arteries, and more force is required from contraction because of this, so a bigger muscle is needed, and you can see that on the diagram just here, thicker muscle. The left ventricle is a lot more thicker because it sends blood to the whole body, from the brains to the toes, which needs a very high force and pressure to do this. Therefore, the wall is a lot thicker, and what you've got to understand is it's not to send more blood. That's not what it's about. It's about sending it at higher pressure. As you can see from the diagram here, the left ventricle sends the blood to the head and the body, which is a lot further than sending the blood from here to the lungs, which are very close to the heart. So these muscles and walls are thicker to send the blood with higher pressure further distances, but not to increase flow. Next objective is to talk about the importance of the septum, which we have already discussed. It separates the left and right chambers, but it also allows pressure to build up in the chamber. Next thing we're going to do is describe the function of the heart and valves. Now I'm going to do this in terms of pressure, using a diagram. So I'll leave this here. So the blood comes through the pulmonary vein, and because it's a vein, it's at relatively low pressure and starts filling the atria and the ventricles here. Now the pressure will start to increase, and as the atria with the thin walls contracts, the pressure will increase to the point where it will push the remaining blood through these valves into the ventricle. Now, when the ventricle contracts, the pressure builds up significantly because this wall is very thick. It can't push back through here because this valve will be closed when this muscle contracts. So this massively high pressure now forces the blood through this valve and around the body. Okay, so we've already talked about valves, and the main thing is that they prevent backflow, but there's a couple of structural differences in that the semilunar valves have flaps, whereas the AV valves have tendons to keep them in place. There are also valves found around the body in veins, because veins don't have high pressured blood traveling in them. Arteries do. Arteries don't need valves, but veins do. So semilunar valves have pockets which basically fill up with blood. As the blood forces through here, you can see it push the little flaps open. But when the blood tries to go in the wrong direction, like it's illustrated here, the flaps are pushed close and it fills up these little pockets with blood here. This is very similar to in the veins as well. Veins and semilunar valves are very, very similar. Okay, well, in terms of the ventricles, we've already talked about this bit, so I'll leave it there. This again is just another picture of an atrioventricular canal defect, and this is where you have a hole between the two ventricles here, and you can see that the problem here is that the deoxygenated blood is mixing with oxygenated blood, and obviously there's problems associated with that. In the exam, all you'll need to do is identify this from a picture. So this is a normal one, and this is the defect.